Good morning, good afternoon, and good evening, everyone, and welcome to this Inside Scientific webinar titled Novel Animal Models and Methods for Studying COVID-19, Golden Hamsters, and Aerosolized SARS-CoV-2 Delivery. I'm Liam Sanyo from the events team here at scientist.com, and I'll be your host today. This webinar has been sponsored by Harvard Bioscience and DSI, so big thanks to them for helping to make this webinar possible. We're very fortunate to have Dr. Valeria Fumagalli and Dr. Nancy Kouassi here, who will present two novel animal models of COVID-19 and highlight recent research conducted using these models. And without any further ado, I'm very pleased to welcome the first speaker, Dr. Valeria Fumagalli. Valeria, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thank you very much. Good morning, afternoon, and evening, everybody. I would like to thank the organizer for giving me this opportunity to present here in this webinar. And today we'll speak about a paper that we recently published on Science Immunology that is about the administration of aerosolized SARS-CoV-2 to K18 human transgenic mice that uncouples respiratory infection from fatal neuroinvasion. First of all, I would like to thank my group, and particularly my colleagues Pietro, Nicole, and Davide that work with me in the BSL3 facility. Of course, my mentor Matteo Iannaccone and Luca Guidotti, foundings, and all our collaborators. Without them, we could not perform this kind of experiment. Moving to science. You know that the most widely used transgenic mouse model for SARS-CoV-2 pathogenesis is the K18 human ACE2, whereby the human ACE2 is expressed under the cytokeratin 18 promoter. And these mice under deep anesthesia are intranasally instilled with a liquid suspension of SARS-CoV-2. And we observed, as others, that these mice experience body weight loss during time, increase in the clinical scores and so in severe disease, and up to 80% of the mice died up to 6 days post-infection, and the remaining one were very lethargic. And we observed that this severe disease was mainly due to infection in the brain. Indeed, we were able to spot SARS-CoV-2 RNA both 3 days and 6 days post-infection in the brain of intranasal infected mice. We confirmed the data by immunofluorescence staining of the nucleoprotein of SARS-CoV-2, and we can observe that almost all the neurons are infected by SARS-CoV-2. And also, we were able to find the SARS-CoV-2 RNA in the olfactory bulb of the mice, both 3 and 6 days post-infection, olfactory bulb that are the center of the olfactory sensory neurons. So we can conclude from this data that intranasal inoculation of SARS-CoV-2 leads to fatal neuroinvasion in K18 human ACE2 transgenic mice. And so all the clinical signs of the disease that we observe are mainly due to brain involvement rather than lung pathology. Indeed, this is not a well model to study SARS-CoV-2 pathogenesis because fatal encephalitis rarely occurs in patients with COVID-19. And this limits the usefulness of these mouse models, hampering study on disease pathogenesis like long-term consequence of the disease or drug discovery and tests of antiviral and vaccine. For this reason, our lab decided to develop a novel model, a novel platform to study SARS-CoV-2 pathogenesis, taking advantage of the aerosolization of SARS-CoV-2 in order to mimic what happened in the human-to-human transmission. For this reason, we take advantage of a nasal inhalation tower system that we obtained from Data Science International, and we are able to nebulize the virus inside this nebulizer head, and the aerosolized virus reach only the nose of the mice that in this case are not anesthetized. And thanks to this model, we are also able to control the pressure inside the tower and the temperature humidity, that are parameters very important to define the dimension and the size of the particle aerosolized. So we set a protocol whereby we are able to nebulize 10 to the 5 TCID50 per mouse in 125 microliters, exposing mice up to 30-40 minutes. And what we observe, we compare the two routes of infection. In red, we can see the aerosol-exposed mice, and in blue, the intranasal infected mice. And we can see that differently from intranasal infection, the aerosol-exposed mice do not experience body weight loss, neither clinical score of the neither sign of disease, neither mortality. Indeed, we did not find any trace of viral RNA in the brain of these mice, both at the 3 days and 6 days post-infection, compared to the intranasal infected mice. However, viral RNA in the lung were comparable between the two routes of infection, meaning that we are able to infect efficiently with aerosol exposure the lung of the Chiatina humanis 2 mice without having fatal neuroinvasion. Next, we were interested to evaluate what happened in the upper respiratory tract. So we evaluated the nose, and in particular the nasal turbinate of these mice. And we were able to detect viral RNA both upon intranasal infection and aerosol exposure, even if at lower strength. But since we were able to find viral RNA in these mice, we were interested also in evaluating if these mice were anosmic, since anosmia is one of the major clinical signs observed in humans. So we perform a social cell discrimination assay, whereby male Chiatina human transgenic mice were exposed to a female bedding or a male bedding inside a cage. In a normal condition, so PBS-treated mice, they should spend more time in sniffing the female bedding, as monitored here, evaluating the sniffing time. However, if mice are anosmic, they should spend less time in sniffing female bedding. Indeed, both upon intranasal infection and aerosol exposure, in particular 3 days post-infection, we have a reduction in the time spent in sniffing female bedding, meaning that these mice are anosmic. This was also true 6 days post-infection upon aerosol exposure. We were not able to perform this kind of assay upon intranasal infection because mice were very lethargic. This is very interesting because we were not able to find, neither in the brain but also in the olfactory bulb, where I said before are present the olfactory sensory neuron, presence of the virus upon aerosol exposure. So we found that aerosol exposure to SARS-CoV-2 leads to anosmia in the absence of central nervous system infection. And how this can be possible? We are trying to investigate this and to understand which is the mechanism. And we perform first preliminary experiment where we perform immunohistochemistry analysis by staining for nucleocapsid protein of SARS-CoV-2, the olfactory bulb where are present the olfactory sensory neuron, and we can see in the upper part of the slide the intranasal infection. Upon intranasal infection, we have that all the neurons are infected, while upon aerosol exposure, as already observed by the level of RNA, we do not have the presence of the virus. But interesting, in the olfactory epithelium, where we have the olfactory sensory neuron, but also cells that are important to sustain the survival of the olfactory sensory neuron, that are two tentacular cells or Bowman gland, we have that upon intranasal infection, a positivity for SARS-CoV-2, both in the olfactory sensory neuron and in these cells that are structural cells. However, upon aerosol exposure, we have the presence of the virus only in Bowman gland. So we are now interested in evaluating how the infection of the Bowman gland can affect then the sense of smells, leading to anosmia. So this is one of the study that we are performing now, but we are also interested in studying the immune response upon SARS-CoV-2. And in particularly, we are interested in studying the immune response upon antiviral treatment to SARS-CoV-2. So we decided to take advantage of our novel route of infection, that is the aerosolization of the SARS-CoV-2, and to test antiviral treatment to evaluate if the antiviral can affect the immune response to SARS-CoV-2. So we take advantage of a protease inhibitor that is an orally available drug already approved, and we treated or not Chiatina humanis 2 mice after the infection with SARS-CoV-2 upon aerosol exposure. And we treated mice 2 times per day by oral gavage, and we evaluate 7 days post-infection in the peripheral blood the immune response. These experiments are still ongoing, so the results are very preliminary. And we observed that in red we have mice treated with antiviral drug, and in blue the mice treated with a vehicle, both infected with SARS-CoV-2. We have here in the first panel at the bottom of the slide, we can see the level of immunoglobulins in the plasma that are immunoglobulin specific for the RBD of SARS-CoV-2, and we have a reduction in the presence of these IgG in the antiviral treated mice. And also we have a reduction in the antiviral response from CD8 T cells, because upon restimulation in vitro, we have less interferon, TNF, and granzyme production. This suggests that the immune response is reduced upon antiviral treatment. And now we are interested in evaluating what happens upon reinfection, if antiviral treated mice have the possibility to generate a memory response and so are protected to a re-challenge of the virus. And this was an example that we use. I would like to give you an example how we use our route of infection to study SARS-CoV-2 pathogenesis. So to conclude, we have compared two different routes of infection, the intranasal inoculation of SARS-CoV-2, that is the widely used, but lead to a clinical picture of encephalitis, pneumonia, but results in death after 6-5 days post-infection, limiting the usefulness of this model. On the other hand, we develop a new platform that is the aerosolization of SARS-CoV-2 with the anosol inhalation tower system that lead to a robust respiratory infection, anosmia, lung inflammation, fibrin deposition, but not fatal neuroinvasion, meaning that the whole clinical parameter that we are describing are mainly due to lung infection rather than neuroinvasion, as observed upon intranasal inoculation. And as very last slide, I would like to show you that we would like to couple this route of infection with new mouse models, since you know that K18 humanist 2 mice is not a very good model because the humanist 2 is under the cytokeratin 18 promoter, so is widely expressed and with a not physiological expression. So we develop an hybridase 2 transgenic mice where only the exon 2 and exon 3 of the murinase 2 are replaced with the human exon 2 and exon 3, because in these two exons were found to be the amino acid specific to bind SARS-CoV-2. And we evaluated that these mice efficiently are infected upon aerosol exposure to SARS-CoV-2, both in the lung, in the nasal turbinate, and not in the brain as expected. On the other hand, since we are interested in the immune response, we decided also to develop a BCR transgenic mouse that express all the B cells with BCR specific for the RBD of SARS-CoV-2. Because we are interested to isolate B cells from these mice, transfer them in K18 human transgenic mice that lack the endogenous antibody production, infect the mice, and see what happens in order to characterize the humoral immune response. And I would like to thank you for to be here and for the attention, and I open to question at the end.
Fantastic. Well, thanks so much, Valeria, for a great presentation. And now I'm pleased to welcome Dr. Nancy Kouassi to the virtual stage. Nancy, thanks so much for joining us, and feel free to take it away whenever you're ready.
Hi. Good morning, good evening, everyone, all from my side, and thank you for the organizer to give me opportunity to speak here today. Yes, my pleasure to present some insight in our work on SARS-CoV-2 pathogenesis in high-risk group and all the lessons we learned from influenza. In our group, we investigate the impact and transmissibility of zoonotic virus like influenza on different high-risk groups. For example, asthmatic patients, obese people, or also pregnant women and children. We identify pathomechanism in human that could, for example, mediate increased severity, and study this mechanism using different animal models like mice, guinea pigs, ferrets, and hamster in our BSL-2 or BSL-3 lab. As a next step, we then translate our findings back from the animal to the human cohort. It's known that sex, age, and also gender have an impact on incidence and severity of several infection disease. In fact, female in reproductive age are more likely to be hospitalized, but also to develop severe influenza infection than male. In our lab, we could confirm that female were more likely to die upon avian H7N9 and also H5N1 influenza A virus infection than male. Furthermore, we could also show a protective effect of testosterone during an infection with the 2009 pandemic influenza virus in female mice. All these data taken together show the importance of sex hormone in infectious disease. In 2019, when the COVID-19 pandemic hits the world, we decided as a whole group to switch our focus from influenza to SARS-CoV-2 and use all our knowledge from influenza infection in this new zoonotic virus. It appears more and more clear in the last decades that sex hormones are able to regulate the whole cell immunity and act far beyond reproduction. In fact, intracellular and also cell surface mediated sex hormone receptor may, upon activation, affect the cell-to-cell signaling, but also systemic hormone signaling pathway via the blood systems. Moreover, during the last decades, evidence grew that sex hormone play an important role in lung health. Sex bias and differences are known in various lung diseases, such as asthma, for example, COPD, and also pulmonary fibrosis, for example, highlighting once again how important the impact of sex hormone on immunity and also lung health. In order to study SARS-CoV-2 pathogenicity and also the effect of SARS-CoV-2 infection on sex hormone metabolism, we established in our lab a golden hamster model that mimic the key features of SARS-CoV-2 pathogenesis observed in COVID-19 patients. Therefore, we infected the male and female golden hamster with the same virus dose using a moderate COVID-19 model, and we monitor body weight and lung function over the time. We could observe that during the first 6 days after infection during the acute phase, the weight loss was comparable between the male and the female. However, during the recovery phase from day 7 to day 14 after infection, the infected male recovered more slowly compared to females. In fact, on day 14 post-infection, the infected male still show reduced body weight compared to females. This delay in recovery in male was also associated with increased fibrosis formation in the lung of the male compared to the female after SARS-CoV infection compared to the control treated hamster. Next, we want to assess if this observed delay in the recovery in the male hamster was associated with impaired lung function. Therefore, we measure longitudinally respiratory function of SARS-CoV-2 infected male and female on day 0, 6, 14, and 21 post-infection using a small animal whole body plethysmography from DSI. We have 6 red chambers here in our free lab that we use for the measurement. We measure the lung function in hamster for 10 minutes with adaptation times for 20 minutes for females and 30 minutes for male. Now to the results. We observed that the respiratory function was more severely impaired in infected male compared to female on day 6 post-infection. In both sexes, we could observe that the tidal volume was reduced on the 6-day post-infection, with which persists until day 14 post-infection in male hamster. Male hamster compensate this deficit in the tidal volume with high breathing frequency, resulting in a normal ventilation rate. In comparison, the breathing frequency stay normal in female hamster that show our similar reduced tidal volume as male. But interestingly, in line with previous data on restrictive lung disease, the mid-tidal expiratory flow, EF15, was enhanced in male hamster, but not in female. And this data highlighting the increased disease severity observed in male hamster compared to female hamster. In line with the previous data, infected male hamsters show a reduction in the peak inspiratory flow and increased expiratory flow. The female hamster just show a reduced expiratory flow, but a normal expiratory flow. As all taken together, this data show that a male hamster presents an impaired lung function after SARS-CoV-2 infection compared to females. We then go further and want to assess if the SARS-CoV-2 infection can have an impact on sex hormones metabolism. Therefore, we quantified testosterone and also estradiol level in the plasma of male and female hamster at various time points after infection. We could see that in infected male hamster, the testosterone level dropped 3 days post-infection compared to the level prior to infection. And also the testosterone level started to recover on the 6th post-infection and was fully recovered on the 14th post-infection compared to the control hamster. We could also observe that the lowest plasma testosterone level detected on day 3 post-infection correlate with the highest virus titer in the lung of the SARS-CoV-2 infected male hamster. In contrast, we could observe that the infected male hamster show an increased estradiol level on day 3 post-infection, and this highest estradiol level was also associated with the peak virus titer in the lung 3 days post-infection. We conclude that the observed impaired lung function correlates with a low testosterone and high estradiol level in male hamster. In female, the picture is a little bit different. In infected female hamster, no significant alteration in plasma testosterone level was detected, in line with the very low level of testosterone in young female hamster in general, like in human. But we could also detect very high lung titer 3 days post-infection in female comparable to the detected titer in the male. The estradiol level, however, was reduced after SARS-CoV-2 infection in infected female hamster compared to the PBS group. And we taken together the virus titer were negatively associated with the estradiol level, particularly on the 3 post-infection in female hamster. We could conclude that in female hamster, the impaired lung function correlates with the low estradiol level. CYP91, as known as aromatase, was identified as a major sex hormone metabolizing enzyme that converts testosterone to estradiol. And the CYP91 gene contains predicted binding sites for key transcription factors such as for cytokines induced NF-kappa B, for example, state 1, state 3, 4, 6, and many others. This highlights the critical role from CYP90A1 in sex-specific immunity. In order to study causalities as well as molecular mechanism, we assess if the golden hamster model could provide a suitable model to study the role of CYP90A1 in SARS-CoV-2 pathogenesis. Therefore, first we assess the CYP90A1 protein expression level in unaffected male and female hamster, and here we detect an increased CYP90A1 protein expression in the male lung compared to the female lung. After infection in SARS-CoV-2 infected male hamster, the CYP90A1 expression increased compare to the control hamster, but also compare to the female hamster. We next want to understand if the CYP90A1 dysregulation that we observed before in the lung of hamster is dependent on the complex in vivo settings in hamster, or if this dysregulation can also be shown in vitro, for example. Therefore, we infected human lung cells, or calorie-free cells, with SARS-CoV-2, and as a control, we infected the cells with SARS-CoV or H1N1 influenza A virus. Here we could detect CYP90A1 mRNA expression induction in SARS-CoV-2 infected cells, but not in SARS-CoV cells, and also not in H1N1 influenza virus infected lung cells. These data show that SARS-CoV-2 has evolved a specific mechanism not observed in its ancestor, the SARS-CoV viruses, and also not in other respiratory pathogens to dysregulate the CYP90A1 in the lung, for example. As next step, we want to understand if the CYP90A1 can actually causally act on the overall impaired lung health from hamster. Therefore, we infected female and male hamster with SARS-CoV-2 and treated the hamster with the clinically approved CYP90A1 inhibitor letrozole. And we monitor body weight and lung function all the time. First of all, we assessed if the CYP90A1 treatment can improve the recovery after SARS-CoV-2 infection, and we could observe an increased weight gain of letrozole-treated male compared to placebo male on day 21 post-infection, and this weight was similar to the placebo-treated male hamster. In contrast, female did not benefit from the letrozole treatment. We then want to know if the treatment may also result to improve lung function in SARS-CoV-2 infected male and female hamster after infection. We measure the lung function of the male and female hamster after infection, and here we can observe that in male infected with SARS-CoV-2, the letrozole treatment results in fast recovery in the tidal volume from day 6, and also attain the level of a placebo group on day 21 post-infection. Accordingly to this data, we also see that the brief frequency shown accelerate normalization in the letrozole-treated animals comparable to the placebo-treated animal on day 21 post-infection. However, no major improvement could be observed in the female hamster. We then assessed other parameters in the lung function, and in line with the improved breathing frequency and also the tidal volume, the letrozole-treated male show improved inspiratory time, similar to placebo-treated animal on day 21 post-infection. And also, like the parameters before, the letrozole treatment did not improve the lung function of the SARS-CoV-2 infected female at any time point. We could, with our letrozole treatment, we could improve the lung function of the hamster at day 21 post-infection. Just to sum up this part, normally in hamster or in male, we have an increased testosterone level and low estrogen level. After SARS-CoV-2 infection, we observe a drop in testosterone level and increased estrogen level in male hamster. After the treatment with the letrozole, we could partially recover the testosterone level and also partially reduce the estrogen level. Furthermore, the male treated with letrozole show improved lung function at day 21 post-infection with reduced fibrosis in the lung. However, the female do not really benefit from the letrozole treatment. Taken together, these findings confirm that the CYP90A1 play an important role in the SARS-CoV-2 pathogenesis in male. Like I said before, we always want to translate our finding from animal models back to the human cohort. And since we identified that an increased expression in the enzyme CYP90A1 is associated with increased disease severity in male hamster, we want, of course, to assess if these changes in these expressions in CYP90A1 expression could be also detectable in tissue from patients who die from COVID-19. Therefore, we analyzed autopsy material from the lung of men and women who were positive for SARS-CoV-2 by qPCR and also died from COVID-19 at 3 independent study sites in Hamburg, Tübingen, and Rotterdam. And as a control, we analyzed the lung material obtained for men and women who were never diagnosed positive for SARS-CoV-2 by qPCR. And here we can see that in all 3 study sites, the CYP90A1 was abundantly expressed in the lung of COVID-19 male patient compared to non-COVID-19 male control at the time point of death. And this was also despite the fact that in COVID-19 male, like we see here, the three images, the white background, the COVID-19 male SARS-CoV-2 RNA was no longer detectable at the time point of death in the male COVID-19 patient. We confirmed this finding by quantifying the mRNA expression level of CYP90A1 in the lung of the male patient who died of COVID-19. And we could also show that the male showed increased CYP90A1 expression in the lungs at the time point of death. However, it's important to say, due to the overall lower female COVID-19 patient, also death case, the conclusive interpretation in female is very limited here. Now to the presentation app. We could show that SARS-CoV-2 infection leads to increased inflammation in the lung of male with increased fibrosis formation and also impaired lung function. We could also show that the high virus titer correlate with low testosterone and high estradiol level in the male in the acute phase of infection. All these data together suggest that SARS-CoV-2 infection can induce several metabolic hits in male hamster, and the first hits occur in the reproductive organs, and the second hit in the lung, resulting in a massive sex hormone dysregulation in COVID-19 male. This may change the hypothalamic pituitary gonadal axis, the HPG axis, to the hypothalamic pituitary lung gonadal axis. Now I want to thank Professor Gabriel to give me the opportunity to work on this great project, and to thank Dr. Schneider-Bertram, Dr. Beck, and also Dr. Schaumburg. We work all together on this animal experiment and all the data that we showed before, as of today. The whole group, our collaboration partner in the UKE, also in Tübingen and Rotterdam, and also the funding and you for your attention. Thank you.
Excellent. Well, thank you, Nancy. Really great presentation, interesting stuff. And with that, I'll just bring Valeria and Nancy back onto the audio line, and we'll kick things off with an interesting question here for Nancy, so why did you choose to perform the experiment in these Syrian golden hamsters and not in mice? Can you provide maybe some pros and cons for this animal model?
Oh, yeah, of course. For example, Valeria showed the humanized mouse model. There are also pros and cons from this model, but we use the SARS-CoV-2, the hamster model, for several reasons. The first reason is the S2 receptor, because maybe most of you know the SARS-CoV-2 virus need the S2 receptor in order to get into the cells. And hamster have the S2 already on the cells, what mice didn't have. That's the first important things here. But also, hamster have the ability to reflect the human data. For example, this is the age bias or the sex difference that we see in human cohort. We can also apply it on hamster, for example, that may are more severely affect for SARS-CoV-2, or also older hamster is also affect for SARS-CoV-2, for example. Also, in hamster, we have able to study the longitudinal approach, long COVID, like I would say, because we have the moderate COVID-19 disease. We can the hamster can they get ill, but after 2 weeks they recover completely, and we can then also measure lung function, for example, or maybe other brain-related dysregulation, for example. We can also study it in the same animals, and also, of course, the whole we have the hamster can have the virus in the whole body, and this is really important to study, for example, drugs. And that's really similar, for example, to human. This is just 2-3 things that make hamster the golden standard for SARS-CoV-2 study.
Yeah, thanks. Great answer there. All right, Valeria, this one will be for you. So thanks for the great presentation. This person says, can you share your experience with animal handling in the system restrainer, and how was the acclimation process?
Yes. Basically in the tower we have a collar that is positioned between the base of the mouse skull and the shoulder in order to avoid thorax compression and maintain normal breathing, since differently from intranasal infection, these mice are not anesthetized, and their behavior inside the restrainer depends on if the mouse is big or not, but we have different dimension of the collar. But we have noticed that if the mouse is bigger, is better, because so for instance the best size is a mouse around 12 weeks of age, in order to maintain the pressure and everything's fine inside the tower. And about the acclimation process, the mice are very quiet. They stay well, I think, because they think like 10 minutes and then they do not move anymore. And the tower, we put the tower inside the hood because we are using the SARS-CoV-2. And what I did is to switch off the light in order to acclimate better the mice.
Fantastic.
I hope to have answered your question.
Yeah, great answer. This is actually a nice follow-up question to that. So Nancy, you used an indirect measurement and used unrestrained animals. So I'm just trying to find where the question went. Yeah, on unrestrained animals, can you name some advantages of doing this as opposed to something more like what Valeria described?
Oh, yeah. I mean, there are pros and cons, of course. The first problem with restraining hamster, they don't want it, they don't like it, and they are really stressed. They're really stressed. They're not they are bigger than mice, but they're also really stressed. And we tried to restrain a hamster. It didn't go so very well. then we think, okay, maybe we have to think about other methods. I mean, it's possible to measure lung function directly, the endpoint measurement, use tracheotomy. Also, you have to kill the hamster or the mice and then measure the lung function directly. But the problem with this method, of course, we cannot measure the same animals from before the infection until 21 days, for example, after infection, like we did in our study, for example. But we can consider to use other systems in order maybe to have the time point, for example, 6 days post-infection, just to measure the one time point. But of course, we cannot measure because it will be a lot of animals. Then we have to kill to have the same data. Yeah. There are pros, of course, but also contra. Yeah.
Yeah, certainly. All right, here's an interesting question for Valeria. Did you get a chance to look at the survival rate of the HY-ACE2 mice? Does the infection actually resolve completely?
Okay, yes. I arrived up to 6 days post-infection, not more yet, because it's a recently generated model, but mice survive whole, as also K18 mice aerosolized, I have to say. But the thing is that the hybrid ACE2 have the expression of ACE2 that is 10 times less in the lung compared to K18 mice. Indeed, the infection and the viral titer and also the PFU that we measure in the lung 3 days post-infection of these mice are fewer than the K18 mice. So I think that yes, the infection resolved completely, also because 6 days post-infection, I already see antibody in the plasma and not viral titer in the lung. But I have to say that it is a mild infection, milder compared to the K18 mice. That I think that is resemble what happened to humans, because the humans that are experience a severe disease are the ones that have comorbidity. So the vast majority are asymptomatic. So I think that we are recapitulating that kind of patient.
Yeah, great point. Okay, here's a good question for Nancy Kouassi. So this attendee says, Dr. Kouassi, thanks for the great presentation. Are you considering repeating this study with a different animal model?
Yeah, this is a great question. We are thinking about it, but to be honest, now at this stage, hamsters are the golden standard to study SARS-CoV-2. To be honest, but of course, for example, to study transmissibility, for example, we can also use ferrets. For example, they are really good models in order to study, or guinea pigs, for example. We can think about it. This is a nice question, but at this time point, we didn't plan to do the same study on different animals. But this is a good question. We also think about it. Yeah.
Yeah, perfect. Okay, here's an interesting one for Valeria. Can you conclusively say that the transgenic ACE2 mice challenged with SARS-CoV-2 die from specifically the neurological infection and not something like pneumonia?
I feel to answer yes, because we observed that even if we reduce the amount of virus with intranasal infection, we observed that like 30% of the mice died and have or were very sick, and the ones that are very sick have a huge viral RNA in the brain. And also we try to play with the viral titer upon aerosol exposure, so we try to increase in that case, but even if we increase the viral load of aerosol exposed mice, we do not experience brain invasion, neither mortality. And moreover, we also perform whole body plethysmograph on both intranasal infected mice or aerosol exposed mice, and we observe that upon intranasal infection 5 days post this infection, we have that all parameters that we can detect with the whole body plethysmograph are affected, while upon aerosol exposure, only the RPF, that is a marker of bronchoconstriction. So since mice are very lethargic, I think that all the parameters that we are reading with whole body plethysmograph in intranasal infected mice are mainly due to brain infection rather than a lung pathology.
Yeah, really good point. Okay, this next question has to do with the sex differences in COVID-2 infection. So Nancy, does letrozole work in castrated hamsters, do you think?
This is the really, really great question. We never checked, but I will just by feeling say not really, because I mean we need a substrate. We need testosterone in order to convert to estradiol. And I mean, castrated hamster are more likely like females, we'll say, although male castrated hamster are like females. I think the letrozole treatment, I don't think so. I will say no. I will say no. But we don't check yet. But I will say by feeling say no, because you don't have the testosterone at the beginning. Then we cannot see the drop in the testosterone, for example, or the increase in estradiol, because we don't have the substrate at the beginning. We don't have a testosterone at the beginning, and we cannot see this drop, and maybe we can all not see dysregulation in the CYP19A1 in the lung, for example, or in the testis. I will say no.
Right. Yeah, that makes a lot of sense. All right. This attendee has written, "Dear Dr. Fumagalli, thanks for the great presentation. May I ask what was the or how were you measuring the amount of virus that reached the lungs when administered via the inhalation tower, and what kind of vehicle you used?"
Okay, starting from the second, we use as a vehicle, we dilute, depends on the initial stock concentration of the virus, because we growth virus in the MEM 0% on Vero A6 or Vero human tempers 2 cells. And if I have to dilute the virus, I dilute it in PBS. But based on that, I also nebulize a control mouse with the same vehicle to then standardize all the analysis that I will perform later. And about how I measure the amount of virus that reach the lung. Okay, this means the deposited virus. Okay, I don't know. The only thing that I can measure is the viral RNA when I sacrifice the mouse, and indeed I saw that the viral RNA is comparable between the two routes of infection, because also with intranasal instillation, I could not know which is the amount of virus that reached the lung, starting from the one that I instilled in the mouse. So it's the same problem. And also what I can control is the amount of virus that is inhalated by the mouse, because I can calculate how much virus I put inside the tower. But also in that case, we have to take in consideration that not all the virus that I put inside the tower, that is aerosolized, reach the lung of the mice, because we have to consider also the exhalation of the mouse. So the only thing that I can say is to play with the dimension of the viral particles, because if we reduce the humidity inside the tower, we can reduce the size of the viral particles, and so hope that this smaller aerosol can go deep in the lung and not remain in the nasal turbinate, for instance. But we could not, I could not say you which is the amount of virus deposited in the lung.
Yeah, makes sense. Valeria, another question for you. We've got actually a few about the aerosolization process. So can you speak at all to whether the aerosolization process reduces the infectivity of the SARS-CoV-2 virus, or if it destroys or damages the viral particle at all?
Okay, this was one of my fear at the beginning, because I was not sure that the virus stay alive upon the aerosolization. But again, since given the same starting concentration between the two routes of infection, and at the end I see the same viral RNA in the lung, I can say that all the viruses that I nebulize stay alive, so remain vital.
All right, very good. Okay, Nancy, so considering the whole body, considering the whole body plethysmography pulmonary system, which endpoint parameter do you think was the most important or telling? Do you think it was the lung resistance or compliance measured? And if not, do you feel it's needed?
Oh, yeah, this is also a very great question. I mean, for us, for example, during the infection with SARS-CoV-2, for example, in male, we could also per eye see this increased breathing frequency. For example, it was really nice to see that what we see per eyes, we could also confirm with the whole measurement. And we measure a lot of hamster, male and female, and we could confirm each experiment this measurement. I think for me, for the whole body plethysmography, I mean, the tidal volume, the frequency were really important, and also this EF50, the bitidal flow, was also really important. On the other hand, will be in the future already nice to have, for example, the resistance in the compliance measured directly in the hamster. It's not possible to do it with the indirect method, but will be great maybe to have a smaller experiment just with a few animals and perform endpoint measurement, for example, for resistance and compliance, for example. For example, the prior the infection, day 6, for example, and maybe day 14 after infection. Will be nice to have, but yeah, it's an animal application to have also. But yeah, will be nice to have. But I mean, in our hand now, to have the possibility to have this measurement, because we measure a lot of measurements. I think we measure 12 parameters, if I remember correctly, and I just show a few of them, and all the data correlates together. I mean, the data are really consistent through the experiment, but also through the animals. It's really convincing that we are doing this well. Yeah.
Excellent. Yeah, that's really great to hear. I think in the interest of time, we'll just have one last question here. So it's about the use of these models for long COVID. Obviously, long COVID, as we get deeper and deeper into it, has been the subject of a lot of study. And we'll start with Valeria. Do you think this animal model, the one that you've been using, do you think it would be an appropriate model to study long COVID?
Yes. I think yes, because mice stay alive that did not die, and also we checked 20-day post-infection, where of course virus were clearated from the lung, so there was no trace of the viral RNA in neither nasal turbinate, neither in the lung, but we were able to find specific CD8 and CD4 T cells, as well as the lung were very damaged. So we have thrombi, hyaline deposition that are patterns that are similar to what observed in COVID-19 patient in the lung of the patient. So I think that it could be a useful model to study also the long-term consequence of the disease. Also because a recent result that I obtained is that I was able to spot inflammation in the spinal cord of these mice that were infected with aerosol SARS-CoV-2. And this is interesting because the spinal cord then is colligated with the central nervous system. So maybe this can explain also some headache and the problems in the central nervous system without having an infection that is observed also in patients. So I think yes.
Excellent. Yeah, great answer. And Nancy, handing things over to you, and maybe research already has been done, but can you speak to whether these golden hamsters would be a good model to study long COVID?
Oh, yes. Yeah, of course, the answer is yes. Because as I mentioned before, the hamster can mimic the human data. It's great to have the sex bias, but also the age bias, but also the obese bias, if I can say so. Then we can, with the hamster model, we can study the long-term effect on different model, male, female, young, old. We can do a lot of things with hamsters. It's really, really impressive. And we can study the immune system, the cytokines response. We can study lung function longitudinally, 40 day, 21, 1 month, 2, 3, 1 year. We can do pretty much anything, because I mean the hamster, we can have the hamster for 3 years, for example, in the lab. Then we can measure all these measurement prior to infection, 20 days after infection, but also 1 year or 2 years after infection, or 3 years after infection. It's a great model. In my opinion, it's really, really great. And we can do a lot of things. And I mean, the people out there also realize the hamster is the golden standard, and a lot of people are working now on all these different high-risk groups in hamster. Yeah, my answer is definitely yes.
Fantastic. Well, for both these models, I suppose that then we'll certainly see in the coming months and years more and more data coming out all the time. So I guess it's exciting to look forward to that. But yeah, just to wrap things up, I mean, Nancy and Valeria, thanks so much for all the insights you've shared with us today. It's been a real pleasure having you with us.
Thank you. You too. Thank you.
Thank you. It was a pleasure.
All right, excellent. Yeah, big thanks also, of course, to the audience here for participating. And last but not least, we'd also like to thank the sponsor, DSI and Harvard Bioscience. So in closing, we hope you enjoyed this Inside Scientific webinar, and we'll see you again next time. Have a great day, everyone.
Thank you.
Thank you.